Okay, hello everyone. So we are continuing our discussion right now on cells, cell contents, and the associated functions. And we actually pulled a few slides from last Friday's class, which we didn't have time to cover, so we will cover them in this capsule, a few of them at least. So here what we have is an evolutionary tree, okay, which branches showing the historical or evolutionary relationships between different groups of organisms, okay, how they evolve over time and how they split and form new species and eventually new groups. And what we have, if we look at all life on Earth, is three domains. Very large groups of bacteria, archaea, and eukaryota or eukaryotes. And the bacteria and archaea together form the group of prokaryotes. You remember prokaryotes are small cells, relatively simple, with very few complex machinery or less complex machinery compared to eukaryotes, right? Eukaryotes are much bigger, they're very complex, diverse organelles within them that perform various functions. Okay, so that's the three domains and the two types of cells and the two groups of organisms, prokaryotes and eukaryotes. Now, we're more interested in eukaryotes in this course, at least. And if we look at eukaryotes, we see that many eukaryotes are unicellular. Okay, so that means one cell is one organism. Lots of protists, little protozoan, phytoplankton, so various small organisms that are made of just one cell. But we also have in eukaryotes multicellular organisms that are made of many cells. So already the eukaryotic cell is complex. But now, even more than that, the organism is very complex because it's made of many different cells, each with different functions that are working together. So what kind of multicellular organisms exist? Well, we have the group of animals, so all multicellular organisms. We have the group of plants, also multicellular. We also have the group of fungi, where we have some multicellular organisms, but we also have some unicellular organisms. In this course, we're going to be interested in animals and plants mostly. Okay. So looking at animal cells and plant cells, we can see that they have several structures in common. For instance, they both have a cell membrane or plasma membrane, and actually all cells have a plasma membrane, right? So that membrane allows movement of substances in and out, and it allows the cell to interact with the outside environment as well. We have nucleus, right? So to store genetic information, the plans to build and maintain the cell. We have ribosomes, very important for protein synthesis. And you know that proteins are all those little machines inside the cell that do most of the work, or all the work, pretty much. The cytoskeleton. Cytoskeleton is found in both animal and plant cells. And the cytoskeleton is a bunch of ropes of protein threads or fibers that are connecting the different organelles, holding them all together, and also so providing some structure to the cell, both in plant and animal cells, and also allowing substances to be transported more easily along those cytoskeleton structures. We also have the mitochondria. So the mitochondria is found in both animal cells and plant cells. So here you see two mitochondria. See one here in the plant cell. Okay, so they both have that. And mitochondrion, or mitochondria in plural, is involved in energy processing. And specifically, the mitochondria is going to make large amounts of ATP, allowing the cell to perform the various tasks it needs. So the ATP is like a little energy packet that we use for chemical reactions. Now, if we look at plants, we see that they have a few features that are not common in animal cells. So one of them is the cell wall. So you can see here in green. And the cell wall, as you can imagine, will provide some protection and structure to the cell. The cell wall is made of a substance called cellulose, which is actually a chain of glucose molecules. So ultimately, the cell wall is made of glucose. And just an interesting trivia, when you think of wood, uh, what is wood? Well, it's actually um, dead plant cells that have their contents are empty, and the only thing that remains is the cell wall. So the wood is basically just many different many cell walls that are attached together and form that structure. Okay, so there are also, in a tree, for example, well, there are some woody parts made of the dead cells, but obviously there are also some cells that are alive in that tree. And finally, if we look at plants structures that are found not found in animals, we can look at the chloroplast. A chloroplast is going to be very interesting to discuss, also involved in energy processing. So in this case, these ones make glucose. So in the next two slides, we're going to look at the mitochondria and chloroplasts. These are our main interest at the moment. So if we look at the mitochondrion, so that's the singular version, and mitochondria is the plural. So for some reason, we often use the plural version. It's present in all eukaryotes, or almost. There's a couple of exceptions, but for the most part, all eukaryotes would have the mitochondrion. It's the main site of ATP production. And so we're going to discuss this in more length in a couple of weeks. But just to give you an overview, how do we make ATP inside the mitochondrion? We use the process called aerobic respiration. And so this is the equation, if you'd like, of aerobic respiration. It's actually a series of many different chemical reactions taking place one after the next. But overall, the overall process is shown here. Sorry, I got mixed up here. Yeah, so you see we're going to use a glucose molecule or some other nutrient. We could use fats, for example, or even amino acids. But typically, the most, an efficient way to perform aerobic respiration using glucose. We're going to use oxygen as well, and we're going to have ADP and P. So what is ADP and P? So ADP means adenosine diphosphate, that means 2, and P is 1 phosphate, okay? So we have the glucose molecule here, glucose. The ADP and P, well, this is the ATP molecule. So the ADP would contain only 2 phosphates, and the P would be just this one here, okay? So we're taking glucose, we're using the energy from glucose, the chemical energy that is found in the bonds of glucose, and we're going to transfer that energy to form a new bond, a third bond to make ATP, right? So we're going to take ADP and P and join them together, form a new bond, and make ATP. In the process, we're also going to make carbon dioxide, CO2, and water. Okay, so then ATP will be used to perform chemical reactions in cells. So ATP, you can think of it as like a battery. And when it's in the ADP state, so I had thought this would play. Oh yeah, so now it's playing. So ADP can be recharged by adding one P and one bond here to form ATP. And then that ATP can, that little battery, now that it's charged, you can plug it in some chemical reaction, and you break the bond and release the energy from that bond. Okay, so what we're doing really, we're transferring the energy from glucose to the bond here in ATP, because that's the convenient, or that's the only way really to use that energy afterwards in small packets for chemical reactions. Okay, so that's aerobic respiration. Now, if we look at the chloroplast, it's also involved in energy processing. Chloroplast is found in plants and a few other eukaryotes, but it's not found in animals and it's not found in fungi. And what the chloroplast does, it makes glucose. Okay? And it uses the sun's energy for that. So the general overview of the photosynthetic process is that we're taking CO2 for the carbon here. We need the carbon. We need the oxygen. We're taking water. We need the hydrogen and oxygen. And we're using the sunlight's energy. So we're going to consider this to be kinetic energy, right? Think of those photons that are arriving and they hit some molecules in the chloroplast, causing some events taking place. But we're going to use that sunlight energy to form bonds in the glucose molecule. Okay, and in that process, we're also going to release O2. Okay, so we're going to convert CO2 and water to glucose and oxygen. And so we're going to require many different chemical reactions, so several steps to reach that, but that's the process of photosynthesis. Okay, so we're basically transferring the energy from the sun now into chemical energy, into the bonds of glucose. I'll just show you again the previous slide. Here, these are the bonds of glucose. It's just a different model of the glucose molecule. Or here, right? So we're between the carbons. These are the bonds that will contain the energy in that glucose. Okay? But then you might want to think, well, what do we want to do with glucose afterwards? So how will glucose be useful to a cell? So maybe you want to pause and think about how any cell, eukaryotic cell, would use glucose, plant or otherwise. How are photosynthesis and aerobic respiration related? So that kind of relates also to the first question to some extent. So what's the link between the two? And last question, how photosynthesis and aerobic respiration affect the global carbon cycle? This we will discuss on Friday, I believe. But now looking at question 2, how photosynthesis and aerobic respiration are related. So let's move forward. So now we're looking at the link between photosynthesis and aerobic respiration. And so we can imagine we have a plant cell. So the plant cell has both chloroplast and mitochondria. Okay. So the plant is capable of producing some sugars, right, glucose, using the light energy from the sun together with CO2. So it requires CO2 and water and light energy, making sugars, as well as producing some oxygen. Now, what can it do with those sugars, that glucose? Well, it could use that glucose then to convert the chemical energy in the glucose to chemical energy, but now in a different molecule that is more useful in terms of being able to take advantage of that energy that is stored in the bonds, So we're going to convert the energy from the bonds in the glucose to bonds to recharge our ATP batteries, right? So we can make energy, or we're not making energy, we're converting energy in a more usable form. And then that energy can be used to perform chemical reactions, right? It's like a small packet of energy. We wouldn't be able to plug in the glucose molecule into a chemical reaction to use its energy, but we can plug in an ATP into a chemical reaction, and that will produce just enough or release enough energy when the bond is broken to trigger the reaction to take place. So if you look at this process, you see that ultimately all the energy comes from the sun, right? But we can convert it in a form that will be beneficial to cells. Interestingly, when you're performing aerobic respiration, you're also going to produce CO2 and water. And now that CO2 and water is useful to make more sugar and oxygen, right? If we have light as well in the chloroplast. Okay, so it's kind of a cycle. And well, you can think also, what else could I do with the sugars apart from recharging my ATP batteries? So other roles of sugars apart from being used in aerobic respiration. So you can think about that as well. So that's the process. Now, if we look at this title here, I like it. Ancient slaves make the world go round, right? So these are really processing the energy and transforming it into a useful form. But why are they ancient? So let's have a further look at the mitochondrion and chloroplast. So those two organelles are a little bit special. They have some features that are common to both organelles, but not found in others. So one feature they have is they have both an outer and inner membrane, so a double membrane. So that's peculiar, right? Special. Another feature they have is they have genetic material. So other organelles don't have genetic material apart from the nucleus. They have their own ribosomes. So the ribosomes that you find in the mitochondrion or chloroplast are different from the ribosomes of that you find in the other parts of the cell. They have their own ribosomes. They have similar enzymes. So that's also interesting to think about, right? How's that possible, or why is that so? And finally, something that might give you even further hint is that they are formed, right? So when a cell divides, or whenever you need more mitochondria or more chloroplasts, these organelles can divide themselves and make new organelles by binary fission. Interesting. So it's a little mystery here. Let's try to solve it. If we were to take the genetic material that you find in the mitochondria, that you find in the chloroplast, and the genetic material that you find in the nucleus, and you were to compare that genetic material, maybe you're comparing a ribosomal protein, for example, which is kind of universal. So in any case, you would see that the genetic material you find in the mitochondria is most similar to genetic material that you would find in some types of bacteria that we call aerobic purple bacteria. So that's interesting. If you look at the genetic material of the chloroplast, it would be most similar to genetic material that you find in some other groups of bacteria that we call cyanobacteria. Okay? And interestingly, cyanobacteria are capable of photosynthesis, right? And the aerobic purple bacteria, they're capable of aerobic metabolism, right? That's why they're called aerobic. While if you look at the genetic material that you find in the nucleus, then you would compare it to other organisms, and it would show on the branch of plants within eukaryotes. Okay, but those two organelles seem to be more similar to prokaryotes, to bacteria. So how do we account for that? Well, through quite a bit of research, eventually a theory was developed. And remember, a theory in biology or in science, it's a well-founded set of hypotheses, right, that are quite solid. So we have quite a bit of evidence, as you know, the evidence that was mentioned a couple of slides before, such as binary fission, the genetic material, and the similarities with the genetic material of bacteria. So the endosymbiotic theory says that there were actually events where one bacteria, yes, was eaten or engulfed by another cell, and that bacteria remained over time and became the mitochondrion. And we had another event where we had a so first we had our proto-eukaryotes, ancestral eukaryotes, that had a couple of organelles. That proto-eukaryote maybe was most similar to an archaea. That's possible. Okay, but in any case, it ate up a aerobic purple bacteria, but didn't destroy it. So that aerobic bacteria remained in that proto-eukaryote, and over time, over many generations, they became inseparable, right? So they became symbionts, right? So symbiont means living together, and endo means inside. So now we have an endosymbiont, which became the mitochondrion. Same thing for the next endosymbiotic event, where we had a cyanobacteria that was eaten up or engulfed by that eukaryote, which already had a mitochondria by then, and that cyanobacteria, over many generations, eventually became an endosymbiont as well, the chloroplast, right? And now again, they're inseparable. So if we look at the evolution of those of the different domains, you can see we have the archaea bacteria here. We have the eubacteria, which means the bacteria, and we see the ancestral eukaryotes. So there was one event where a purple bacteria was inserted, right, into an ancestral eukaryotic cell, such that now all or most of the almost all eukaryotes carry a mitochondria within them, within their cells. There was an event here, or possibly several events, where a cyanobacteria was engulfed into an eukaryote. By then, the eukaryotes had already diversified, so that became the chloroplast, and that chloroplast is found in plants as well as a couple of other groups of organisms as well, right? It entered the ancestor of plants plus those other groups. All right. While animals and fungi will not have chloroplasts. So hopefully that makes some sense. And I'd like to finish with a link here to geology. So for some of you, that might be interesting. So on this slide, we're going to look at the changes in atmospheric oxygen and evolution. So if we look on the graph here, we have Earth formation around 4.5 billion years ago, and pretty soon the earliest life forms appeared. These would be simple prokaryotes. And among those prokaryotes, most of them were anaerobic. What does that mean? Anaerobic means that they make ATP without O2. They're not using oxygen to make ATP. So they're not performing aerobic respiration. So most of those organisms would be anaerobic. Well, there was very little O2 in the atmosphere early on, so there was no possibility to use O2. Okay, but eventually the oxygen in the atmosphere gradually increased, and over time we also had the evolution of aerobic prokaryotes, and in this case they make ATP using oxygen, right, in the process that we have discussed, which is happening in the mitochondria of aerobic respiration. So we had prokaryotes that were anaerobic and a few that were aerobic. But most of them anaerobic. Again, there was not lots of oxygen in the atmosphere, and oxygen is actually toxic to many of those anaerobic organisms. Okay. So in any case, that's the early life forms. Eventually, there was evolution of some prokaryotes that were able to use to make glucose by photosynthesis, right? So these were the cyanobacteria. And so as soon as this process became evolved, the cyanobacteria started to become more and more common, and they produce more and more oxygen. So the oxygen increased in the atmosphere. But early on, it didn't increase that much because most of that oxygen was oxidized. There were lots of iron in the Earth crust. There is a lot of iron, so the oxygen was binding to the iron to form rust, and therefore it was not going into the atmosphere. But eventually, when most of the iron was rusted, right, oxidized, around 2.4 billion years ago, now the cyanobacteria were still producing oxygen, and then that concentration of oxygen increased relatively rapidly within 200 million years. Okay. In the meantime, it's important to realize that we also had some sort of ancestral eukaryotes, similar to what we've seen on the previous slide, a proto-eukaryote that had some organelles, some more complex structure compared to the prokaryotes, but it didn't have a mitochondria yet. Now, when that oxygen concentration increased between 2.4 and 2.2 billion years ago, well, that generated a very strong selective pressure because the anaerobic organisms were actually not favored, right? Oxygen can be toxic to them, so they would not survive very well. On the other hand, the prokaryotes that had evolved the ability to use oxygen to make ATP, well, were using up that oxygen, so it was not too toxic to them, and they also had that, much more energy available to them than others. So then they became much more common. Okay, so these aerobic prokaryotes, they were making ATP with oxygen. So that selective pressure was very strong. So it's not surprising that we had this endosymbiotic event where a anaerobic eukaryote, which was not doing very well at the time because there were increasing amounts of oxygen in the atmosphere, but that eukaryote got lucky and ate or engulfed an aerobic bacteria, which was able to detoxify the oxygen that was getting into the cell and use it for energy purposes, right? So this is when we had our first endosymbiotic event. So you can think here that the selective pressure was quite strong, and the benefits of having that aerobic bacteria inside was high. And so we had our first endosymbiotic event, okay? And by then, by 2.2 billion years old, billion years ago, we had our eukaryotes had evolved, and all eukaryotes had, or almost all eukaryotes had a mitochondria, okay, as a result of that event. Okay, and that will be it for this capsule. See you later. Thank you.